Dear learners, welcome you all in the course of Immunology BZYET 141. Basically, it is a elective courses, and this courses for BSc General Program. Dear learner, my name is Dr. Siaram. I am an associate professor, Discipline of Life Science, School of Sciences, IGNOU. Dear learner, today I want to discuss very important topic that is known as the cells of the immune system. It is a very important topic we will discuss in detail. Dear learner, what is the objective of today's sessions? After finishing of this session, you will be able to discuss the process of generation of the immune cell. How to generate the immune cell, we will discuss in detail, like hematopoiesis are there. After finishing of this session, you will be able to explain the regulation of the production of immune cells, how the immune cell will will produce, and we will you will also able to discuss the characteristic and function of the various immune cell. The various immune cells are there, like T lymphocyte, B lymphocytes are there. And after finishing of this session, you will be able to explain the mechanism of the immune response. And after finishing of this session, you will be discuss the properties of the antigen recognized by B cell and T cell. The dear learner, it is a number of the learning outcomes are there. So I want to discuss one by one. So dear learner, basically my session is divided into two parts. In part first, we will discuss the hematopoiesis, how the different type of the cell will produce, how it matures in different organs, and we will also discuss their regulations, how they regulate like apoptosis, which are very important process are there. We will discuss in detail. Another thing I want to discuss in this session, the cells of the immune system, like granulocytes. They have the granules are the blood cells. They have granules like neutrophil, basophils, and eosinophils and mast cells are there. While in part second, we will discuss in the agranulocytes. This type of the cells, they don't have the granules in the cells, like monocytes, macrophages, dendritic cells. And finally, we will discuss in detail the lymphocyte, basically T lymphocytes and B lymphocytes, and we will also discuss the dendritic cells, which are involved in the killing of the pathogens. So these are the two session, part first and part second. So I will discuss one by one in part first and part second. So it is part first. In part first, we will discuss in detail the hematopoietic stem cells. So basically, it is a very important cells. In this cell, they are able to generate the various type of the cells are there, like agranulocytes and granulocytes and lymphocytes are there. So your hematopoietic stem cells, it is a pluripotent cells, means they are able to convert into different type of the cells and differentiate into granulocytes, erythrocytes, mast cells, monocytes, megakaryotes, and lymphocytes. So these are the various type of the cells they are able to generate with the help of the hematopoietic stem cell, which are the pluripotent cells. And HSC is very low population, less than 1 HSCs per 5 into 10 ki power 4 cells. it is not easy to grow in vitro. So we cannot grow in vitro very easily. It require number of the things, then we can grow the different type of the cells are there, like hematopoietic stem cells. They require the different conditions to grow, and HSCs have ability to self renewal, means they are able to renew it very well. And HSC have tremendous proliferation potentials on demands. So according to demands, your HSCs, hematopoietic stem cells, they will proliferate it by T cell, and T cell, and there a lot of population we will a lot of population will proliferate of the different type of the cells on the demands. And HSCs develop and mature in the stromal cells. So basically stromal cells, it is a meshwork. They have the lipocytes, endothelial cell, fibroblast, and macrophages of the bone marrow. So basically the stromal cell meshwork are there. They are able to generate the HSCs, hematopoietic stem cells. It is a very important to grow the different type of the cells into the bone marrow. Basically in bone marrow, the granulocytes, agranulocytes, and lymphocytes, they are able to generate in bone marrow. It is a very important part of our body. It is a it is a you can say the lymphoid organs are there. So in this case the primary lymphoid organs are there. So in this case the cell will generate various type of the cell will generate in bone marrow. And stromal cell have stromal cell are the non hematopoietic. It nurture and growth and differentiation of HSCs. Basically they provide the nutrients, they provide the particular environments to grow the HSCs. Upon infection, activated T cell and macrophage induce hematopoiesis by producing hematopoietic growth factors. So hematopoietic growth factor is a very important factor. They are able to upon need of the cells, upon infection of the cells, the various type of the cells, they will differentiate and they will proliferate in a particular region. Hematopoietic growth factors such as the target cell which diffuse or by binding receptor, it will reach through the diffusion or by the binding of the receptors are there. The various receptors which are available in the HSCs and the progenitor cells, various progenitor cells are there like T cell, T progenitor cell, lymphocyte progenitor cell, and myeloid, lymphoid progenitor cell, myeloid progenitor cells are there, or ultimately they are able to produce the different type of the immune cells. The human produce approximately 3.7 into 10 to power 11 blood cells per day to maintain steady state level in the circulatory system. It is a very important things are there behind the your immune response. It will maintain the steady state. They will produce 3.7 into 10 to power 11 blood cell per day. So ultimately the steady state level it will maintain. Otherwise our body defensive system will fail. And during the hematopoiesis, the cell division and differentiation are regulated by the apoptosis. So apoptosis, it is another very important phenomena are there, or you can say it is a mechanism to regulate the population of the various cells inside to the bone marrow. The failure of apoptosis may cause the leukemia. If apoptosis will not take place properly, the cell will not reduce inside to the body. So ultimately cancer will cause and leukemia will cause. I want to discuss the hematopoiesis, which are very important phenomena of this. Here is a very important graph are there. You can see here is the graph. Here is the prenatal and postnatal. It is the um, cells are there. Here is the um, uh, fetal month. Fetal month. Here is the birth and age of the year. You can see it here. The it start your maturation of the B cell in T cell. It will form into the yolk sac after 2 to 3 months, 0 to 3 months. And it is divided into the three main stages are there. The 0 to 1 month of the embryonic life, cell are formed in mesenchymal and yolk sac, and 0 to 2 month are embryonic life of the cell form into the mesenchyme or yolk sac. So it will start from the yolk sac, it will take 2 to 3 months. And hepatic stage, another stage are there. The hepatic stage, in this case HSCs migrate to the liver from the yolk sac and subsequently to the spleen. And third one is the medullary stage. By 15 month into the bone marrows, it transfer. So in case of bone marrow, the B cell and T cell will form, or ultimately it will mature. B cell will mature into the bone marrow, and T cell it will mature into the thymus. You can say the training will take place of the T cell in the thymus to fight against the pathogens. So you can see it here is it is the your cells, your B cell and T cell will generate into the liver and spleen. After 4 5 months it will shut down, it will seize it, and then bone marrow are taken care of the formation of the B cell, T cell, and other cells are there in case of this bone marrow. In fact, bone marrow of all bones are there. In adult, vertebrae, ribs, sternum, skulls, scrotum and pelvis, proximal ends of the femurs. So it will take place of the formation of the B cell, T cell, and their maturations are there. So it is a hematopoiesis. It start from the fetus to the adults. The in case of fetus, yolk sac, liver, spleen which are involved, and after that in case of the infant, after birth, the bone marrow and all bone which are involved for the formation of the B cell and T cells. In adult, there are the several bones are there. They are able to generate the B cell, T cell, and other cells are there, or ultimately they will mature it, or they are able to um, uh, uh, to fight against the pathogens. And multipotent hematopoietic cells are there. Multipotent hematopoietic stem cells are differentiated into common lymphoid, myeloid or lymphoid progenitor cells are there. The multipotent hematopoietic stem cells, they can differentiate into common lymphoids as well as common myeloid progenitors are there. In the nature and quantity of the growth factor in the microenvironment influence the stem cell differentiation. Basically, in case of the bone marrow, they provide the microenvironment for the differentiation of the hematopoietic stem cells, and lymphoid cells such as the B cell, T cell, NK cell, and some dendritic cells are derived from the common lymphoid progenitor cells. And in case of myeloid stem cell produce RBCs, and many other blood cells are there like neutrophils, eosinophils, basophils, monocytes, mast cell, and dendritic cells, and platelets will form. So there are the two type of the lineage which are generated in the bone marrow. One is the lymphoid lineage, another is the myeloid lineage, or they are able to produce the different type of the cells are there. And I want to discuss the differentiation of the hematopoietic stem cells. You can say here is the hematopoietic stem cells. They have capability to the renewable it will renew very well self-renewable mechanism which are involved with the HSCs. Here is the common myeloid progenitor cells are there. You can see it here is it can convert it into the it can change into the different sections like megakaryocytes, and it will convert into the thrombocytes. Platelets are there, so ultimately they will take in role of the clotting of the bloods. Here is the erythrocytes, mast cells, and myeloblasts are there. In this case, basophil, neutrophils, and eosinophils, and monocytes. Monocytes again convert into the dendritic cells, and macrophages are there. So it is a differentiation of the hematopoietic stem cells are there. In our bone marrow, the number of the cell it will generate through the common myeloid progenitors are there. And here is the common lymphoid progenitors. In this case, the NK cells will be generated. Another lineage are there. It is the small lymphocytes are there. If you compare the size of the blood cell, you can say their size is the very small. So small lymphocytes are there. It is the T lymphocytes and B lymphocytes, and again it convert into the plasma cell and memory cell. Plasma cell, they are able to produce the antibodies. So these are the differentiation of the hematopoietic stem cells. Cell found in the blood vessels and tissues. So you can see it here is the blood vessels are there, like artery or veins are there. Here is the blood vessels. In the blood vessels, the number of the cells which will flow from heart to the heart to the various part of the body or tissues. Here is the NK cells which are present into the blood vessels, and plasma cells, and lymphocytes, neutrophils, platelets, and eosinophils, and basophils, monocytes, and erythrocytes. These are the different type of the cells which are found into the blood vessels. They flow from heart to the other part of the body tissues are there. Another is the tissues. In the tissues, the number of the cells which are present, like NK cells which are available in the bloods as well as to the tissues, plasma cells, and T lymphocytes, and macrophages, and mast cell, and eosinophils. So these are the various type of the cells which are available in the tissues. These are the distributions of the different type of the cells in the blood vessels as well as in the tissues. Hematopoietic inducing environment, it is another very important. This environment is maintain the actual situation where the your blood cells they will generate the myeloid progenitor cells and lymphoid progenitor cells. They are able to convert into the different type of the cell. They differentiate into different type of the cells are there. So here is a very important phenomena are there that is known as the hematopoietic inducing environment. So in these environments, it is very important. They provide basically stromal cells are there in the bone marrow. The stromal cells are a non-hematopoietic hematopoietics. It nurture and growth of the and differentiation of the hematopoietic stem cells. Upon infection, activated T cells and macrophage induce hematopoiesis by producing of the hematopoietic growth factors. So here is very important things are there, like hematopoietic growth factors are there. It will release, or ultimately they will differentiate into the different type of the cells, and even T cell, macrophage, it will proliferate in particular environments. which are available into the bone marrow. In one HSCs can produce 10 to the power 6 mature blood cells after 20 division. So you can see 10 to the power 6 mature blood cells after 20 division. So they have a lot of capability of the hematopoietic stem cells are there. They also affect the differentiation of the HCs by providing the hematopoietic inducing environment with the help of the cellular matrix. The various cellular matrix are there, like the adipose tissues are there, fat cells are there, various environment in bone marrow they will provide it, or ultimately they are able to differentiate into the different type of the cells. When the growth factor are present, HSCs respond, and which increase the production of the other cells. The environment, hematopoietic inducing environment is very important, which are available in the bone marrow. In bone marrow, the number of the cell will differentiate, or ultimately it protect our life from the infections. So without this environment, the cell will not generate, your immune response will not take place. So these are the very important things in case of the differentiation of the cells. You can see it here is the few transcriptional factor essential for hematopoietic lineage. Here is the hematopoietic lineage. They are able to convert into the different type of the cells are there. Here is the different lineage. Here is the factor like GATA first, GATA first, and they are they are necessary for the thyroid lineage. GATA second, thyroid, myeloid, and lymphoid lineage. Here is the growth factors are there, like GATA 2. It is very important. They if your hematopoietic stem cells, they will produce the GATA 2, they are able to convert into the thyroid, myeloid, and lymphoid lineage. PU1, erythroids, maturation stage, myeloid later stage, and lymphoid. So it is another very important factor. BMI1, all them which are very important. It is factors are their growth factors, and they will maintain it, or ultimately they will differentiate into the different lineage. And ECORAS, here is it is very important for the lymphoid. OC2, B lymphocyte, differentiation of the B cell into the plasma cell. So these are the growth factors. Transcriptional growth factors are there, or you can say the transcriptional factors, which are very important for maintaining of the hematopoietic lineage, or it may be thyroid lineage, myeloid lineage, and lymphoid lineage. Another thing I want to discuss here is hematopoietic growth factors are there. You can say here is the stem cells, multipotent stem cells are there. You can say the hematoblast are there. Here is the common progenitors. But in case of the different growth factors are there, interleukin first, interleukin 3, and interleukin 6, and CSF. These are the various growth factors are there. If this growth factor in excess amount, they are able to convert into the common myeloid progenitor. So these are the tightly controlled systems which are available in the bone marrow, or ultimately they are able to produce the common myeloid progenitors. Another is various factors, CSF and TPO, IL3, interleukin 3, and GM-CSF. They are able to convert into the megablast, and finally it will convert into the thrombocytes, or platelets are there. Here is the various growth factors. Another growth factors are there, GM, CSF. They are able to convert to the myeloblast, and myeloblast convert into the various very important growth factors are there. Without this growth factor, the differentiation will not take place. So it is very tightly controlled systems are there, or they according to need of the particular cells are there, they will differentiate into the different type of the cells are there, like basophils and neutrophils, eosinophil and monocytes. And further monocytes it convert into the macrophages are there. Another is the common lymphoid progenitors are there. These are the various growth factors are there, like interleukin 2, interleukin 7, interleukin 12, and CSF. These are the various factors are there. They are able to convert into small lymphocytes like B lymphocytes. And here is the different IL-1 and IL-2, interleukin 6, interleukin 7. They are able to convert into the T lymphocytes. And here is the B lymphocyte, and again the T lymphocyte convert into the plasma cells and B cells, and B cells are able to produce the antibodies. So basically the hematopoietic growth factor which are responsible for the differentiation of the different type of the lineages and different type of the cells are there. In hematopoietic genetic regulation, expression of the lineage specific gene is needed for to the pluripotent stem cell differences into the various cells. The product of these gene regulate stem cells and differentiation. The number of the growth factors I have already discussed it. You have seen they are able to differentiate into different lineages are there. When a specific gene is deleted, the mice cannot produce the RBCs or specific WBCs. This has been studied using the knockout technique. So it is a very important technique. With the help of this technique, we can see the function of the particular growth factors are there. We can disrupt the particular gene, or ultimately we can see the differentiation of the particular cells are there. This particular differentiation of the different type of the lineages will take place or not. So it is a very important technique, and transcription factor have been identified using this technique. So this technique is very important. Knockout, we can knock out the particular gene and we can see the function of the particular gene and particular growth factor in the cells. And another thing I want to discuss, the nexus between the hematopoiesis and apoptosis, production and destruction of the cells in component. So here is nexus is a very important for the hematopoiesis, and apoptosis. Both are very important to maintain the cell counts, which are very important. The cell counts are there. The RBCs has average lifespan of about 120 days. The lifespan of various cell white blood cells and range from few days to for neutrophils in 20 to 30 year, for some to neutrophil to 20 to 30 year of some lymphocyte. To maintain steady state level, the average person need produce approximately 3.7 into 10 to the power 11 WBCs per day. Apoptosis is a regulatory process of involve the activation intracellular proteins, cell shrinkage and condensation chromatins. And this apoptosis is a very important. They can maintain the cell population inside to the body. During hematopoiesis, cell division differentiation are regulated by apoptosis. Failure of apoptosis may cause the leukemia and necrosis and other mechanism in which death of the cells tissues take place. It occurs when there is insufficient blood flow into the tissues. Necrosis is the irreversible. Here is the cellular change. You can see here is the cellular change are there. Here is the normal cell. The condensation of the chromatins are there. Membrane valves. You can see it here is the valving sucker, and apoptosis, apoptotic body will release. Here is the cell fragmentation will take place in normal cells, and you can see in case of the necrosis, normal cell are there, swollen mitochondria and swollen Golgi apparatus and membrane valves, and ultimately the cell will die. Here is the normal adult blood count. normal, you can see it RBC 5 into 10 to the power 6, platelets 2.5 into 10 to the power 5, leukocytes. So these are the cell counts are there. Here is the leukocyte percentage, in neutrophil 50 to 70%, monocyte 1 to 6%, basophils in less than 1%. So these are the, it should be maintained. It is maintained through the hematopoiesis and different growth factors which are involved, and apoptosis which are involved. Ultimately they will maintain the steady state levels of the cells are there inside to the body. Another thing I want to discuss, the separation of the blood cell into the component. Here is the plasma cells are there. You can see it. They contain the protein, ions, waters, nutrient, waste, gases. In case of if you centrifuge to the blood, so you will get the three layers are there. Here is the buffy coat are there. White blood cell and platelets are there. Here is RBCs. The cells of immune system, you can see, I have already discussed it, the various cells which are involved into the innate immunity, non-specific first time defense. There are the various cells are there. I have already discussed it, cells of the immune system. I want to discuss one by one. Here is the granulocytes. You can see it. Here is the structure of the granulocytes, and here is the glycogen granules are there. Here is the phagosomes. Neutrophils, 50 to 70% circulation of the peripheral blood form into the bone marrow from the myeloblast stem cell called the polymorphonuclear cells. Important role in the inflammations, and first circulatory cells to migrate to the site of the infections are there. So one is very important cells which are the granulocytes, that is known as the neutrophils. Another is the eosinophils. Eosinophils, it is a bilobe. You can see the structure of the bilobes and polymorphonuclear leukocytes. Their size 12 to 15 micrometer, 2 to 4% of the circle. It is another very important cells are there. They are able to release the toxic major basic proteins such as the histamine, lymphokines, interleukin 5s are involved in the destruction of the histamine, contain the peroxidase, DNase, lipase, and there are the basic proteins are there. With the help of this, they are able to destroy the pathogens. Another is the basophils. You can see here is the multi-lobe nucleus are there. They basically they are able to produce the histamine, prostaglandin. With the help of this, they are able to kill the pathogens. Another is the mast cell. You can see the structure of the mast cell are there. It is found into the variety of organ, connective tissues, skin, digestive tissues are there. And the mast cell and blood basophil both play important role in the developing allergies. And dear learner, I want to summarize part first into the, you have learned the various thing into the part first. So you have learned the blood cell originate from the hematopoietic stem cell. Progenitor stem cells arise from HSCs, in which further differentiate into the other cell. During hematopoiesis, stem cell differentiate, involved in expression in various gene. And cell of the immune system, you have learned related to the neutrophil, basophil, basophils, eosinophil, and mast cell. Dear learner, we will take a short break. Don't go away. We will back within 2 to 3 minutes. Thank you.
use honey in our everyday life. Normally we buy honey from market, but we don't know the quality of the honey. So we can judge the quality of the honey, whether it is pure or it is adulterated with sugar solution. We can do a simple test at home and you can judge whether the honey is pure or adulterated. For this test, we have to take a glass of water. Now we'll add a spoon of honey. Here you can see the honey is going to the bottom and it is not getting dissolved in the water. So this is pure honey. Now we'll take another glass of water and I'll add another sample of honey. Here you can see honey is getting dissolved in the water. That means this is not pure. This is adulterated with sugar solution. So this is a very simple test to judge whether the honey is pure or adulterated. We all use sugar.
Dear learner, welcome in my part second. In this session, we will discuss about the agranulocytes. In this case, the agranulocytes, the monocytes and macrophages are there. Another is the dendritic cells, and after that we will discuss in detail the lymphocytes. The various lymphocytes are there like B cell, T cells. So I want to discuss one by one. Agranulocytes. According to name, you can see it. Here is the no granules are there in particular cells like monocytes, and 3 to 8% of the circulating WBC and life span in month. So within month it will die. The largest WBC in diameter about around 18 micrometer. If you compare the size of the WBCs, their size it would be the 18 micrometer. It is the largest cells are there, and they are U-shaped. You can see it. Their nucleus it would be the U-shape or kidney shaped. You can see it, and they take the dark stain pale blue colors, and monocytes circulate into the blood streams in about the 8 hours, during which they enlarge and they then migrate into tissues and differentiate into the macrophages. So after 8 hours, your monocytes will circulate into blood stream, or it will convert into the macrophages and dendritic cells. And macrophages and dendritic cell basically the APCs, antigen presenting cells, which involve into the immune response. If this will not take place, the self and non-self mechanism will not be there. And another thing which are very important, I want to discuss the very important disease that is known as the monocytopenia, associated with the deficiency of the excess monocytes in the peripheral blood. If excess monocytes will be there. You can say it is a condition of the cancers are there. Maybe spread of the cancer will take place after the excess monocytes. Another is macrophages. Macrophages is the first discovered by Eli Mechnikov in 1884 and survive only a day or so before the later tissues mature into the macrophages. So first of all, the cells which are released in the hematopoietic stem cells, or it will convert into the macrophages. The monocytes macrophages are APC, antigen presenting cells, and they degrade enzymes. They have the degradative enzymes in the lysosomal granules and chew ingested proteins in present in the adoptive cells. So basically, adoptive immunities are there. They have the monocytes and macrophages. When the monocyte enter into the tissues and become macrophages, they enlarge and increase the production of the intracellular macrophages are there and can live a year in the tissues and highly motile. So these are the quality of the macrophages are there. Macrophages like cell including dendritic cells found in the lymph node and Langerhans cells of skin and glial cells and the central nervous system, they are they are found and name according to their tissues location and functions. One is the alveolar microphage, which are available in the lungs. According to name, you can understand. Histocytes, the connective tissues. Basically, you can say the histocytes macrophages are there. Kupffer cells, which are available in the liver, and mesangial cells, which are available into the kidney, microglial cells, which are available in the brains, and osteoclast, which are available into the. So these are the different type of the macrophages are there. And you can understand here the phagocytosis and exogenous antigen processing by the macrophages. How can exogenous antigen processed by the macrophage? Here is the bacteria are there. Bacteria, it will by the exocytosis are there. Here is the it will form to the phagosomes and phago, and then it will attach it to the lysosome. Phagolysosome will forms. So you can see it here is the various mechanism which are involved, or ultimately they will display it. So your phagocytosis and exogenous antigen processing by the macrophage, and exocytosis and degraded and fragments are there. So you can see it here is the antigen presentation by class second MSC molecules. So in this case here is the MSC class second molecule, they present the particular peptides or particular antigens. So these are the very important mechanism of the macrophages are there. Another, the phagocytic cells recognize the antibodies on the surface of an antigen. Here is the extracellular bacteria, or you can say they have the antigenic properties. Here is the antibodies. It will bind with, or here is the receptors are there. So it is bind the antigen antibody complexes will bind it. Here is the phagocytize, or ultimately it will clear in the macrophages. So these are the mechanisms are there. That is known as the known as the opsonin. Another is dendritic cell, and located into the skin inner layer and nose and lung of the lung and to the gastrointestinal tract. And there are the four type of the dendritic cells are there. First is the Langerhans dendritic cells. Second is the intestinal dendritic cells. Third one, myeloid dendritic cell, lymphoid dendritic cells. All dendritic cell express class second MSC molecule, and B7 is co-stimulatory molecule in high level. Hence, this type of the dendritic cell are more efficient than other APCs. Other APCs like and the B cells are there, they can process it, but it would be different, and they which require a trigger to the function of the APCs. So it would be different, you can say. They have the B7 is co-stimulatory molecules. Another is lymphocytes. The lymphocytes account 20% to 40% of the white blood cell in our body and 90% of the lymphocytes. And they can enter into the tissues, enter into tissues space and lymphoid organs such as well as circulate into the body and the lymph. An immune system characteristics such as variety of the specificity, memory and non-self. So they have ability, lymphocyte have ability, variety of the characteristic of the lymphocytes are there, like they are the very specific, they have the memory, and they have ability to recognize the self and non-self mechanism, non-self antigens are there. So I want to discuss one by one the lymphocytes. One is the very important lymphocyte that is known as the B lymphocytes. And B lymphocyte form and mature in the bone marrow, and antigen specific antibodies are made. It is very important mechanism are there, very specific antibodies which are produced, which are made in the blood system. And 10 to 15% of circulating lymphocytes are the B lymphocytes. And activated B lymphocyte are factory of the producing of the antibodies, like plasma cells are there. You can say it is a activated B lymphocytes. It is the factory for producing of the antibodies, and they produce antibodies which are very specific. You can say they are able to reduce the particular pathogens. And B cell can switch the classes and produce the different type of the antibodies are there, like IgA, IgG and IgM. The class switching mechanism is very important. They are able to switch in the B cells, or they are able to produce the different type of the antibodies against the same antigens with the same specificity. And mature B cell contain 1 to 1.5 into 10 ki power cell 10 ki power 5 immunoglobulin receptors for the antigens. The B cell is essential for the humoral immunity. And another is the T cell, which are very important. They are able to protect our body from the infection. The T cell produce in the bone marrow and mature in the thymus. They will take the training how to fight with the pathogen. Here is thymus is very important organs are there. They will take the training in the thymus how to fight the against the particular pathogens. And they participate in the cell mediated immunity. So basically the T lymphocytes, they are basically, you can say, the T cytotoxic immunity are there. They will take a role in the cell mediated immunity, important in production of the B cells antibodies. And CD4 CD8 is found in on the mature cell. It is very important. Receptors are there, or you can say the particular proteins which are present on the surface of the T cells are there. They will recognize with the particular organisms. And the helper T cells are cell that have to the CD4 markers, and T cell that express to the CD8 marker are cytotoxic cells, and TCR to the membrane transmembrane heterodimer composed of the two polypeptide chain exist onto the each T cells. The mainly three subpopulation of the T cells are there. So one is the T helper cells. You can see here is the T helper cells are there, and T cytotoxic cells are there. And third one is the suppressor T cells, and are a specific T cell involved in suppressing humoral and cell mediated immunity. So that is known as the suppressor T cells. And cytotoxic T cells, and T cell express the CD8. You can see here is the CD8 molecules which are present on the surface of the T cells, called cytotoxic cell. Another is the CD4 receptors are there. CD4, the T cell express CD4 molecule are called T helper cell. They are restricted and recognize only those antigen have MSC class second molecule. So ultimately the CD4 and CD8 molecule, they will recognize the antigens through the self and non-self mechanisms. Antigen presenting by MSC molecule, T cytotoxic and T helper cells are there. You can see it here is the virus infected cell, the antigen fragment which are available in the particular cells. Here is the TCR. The TCR, they have the class first MSC molecule. Here is the T cells. T cells are there. Here is the they have the class first MSC molecules. They have the TCR, T cell receptors. Here is recognized antigen by the T cells through the MSC first molecule. So they are able to recognize the particular pathogen through the MSC first molecule. Another is the antigen presenting cells. APC, antigen presenting cell, here is the class second MSC molecule. It is the T cytotoxic T cell, it is the T helper cells are there. They are able to recognize T cell through the MSC second molecule, or ultimately it will enhance the immune response. Another is the NK cell. NK cell are large granules. You can see here is the large granules are there. In particular lymphocytes, and kill the virus infected and transformed cells, and killing non-specific. Basically they distract the pathogen which are the non-specific. Any pathogen can kill by the NK cells. And lack of the membrane receptor like T and B cell, and target recognition occur by to the killer inhibitory receptor, that is known as the KIR receptors are there. With the help of this KIR, they are able to recognize the infected or tumor target. And NK cell adhere to infected target cells and induce their cell death by apoptotic signals mediated by the perforin. Basically NK cell, they will kill the pathogens with the help of the apoptotic signal. There are the various apoptotic signals are there: perforins, granulozymes, and tumor necrosis factors. So these are the three main factors which are released by the NK cells, or ultimately they will kill it. Here is perforin pore forming cytolytic proteins, serine proteases released by the cytoplasmic granules with cytotoxic cells, and tumor necrosis factors. So these are the various factors which are released by the NK cells. NK cell can also kill through the ADCC, antigen dependent cell mediated toxicity, via CD16 receptors. You can see it here is the target cells. The antibodies, they will bind or the specific cell, tumor infected cells are any virus infected cells are there. Here is the NK cell. NK cell, they will recognize it, or ultimately they will kill the pathogens. So that is known as the antibody dependent. Here is the antibody which are involved in the destruction of the pathogen or killing of the pathogen. So you can say it is antibody dependent cell mediated cytotoxicity by the NK cells. Dear learner, I want to discuss, dear learner, I want to summarize summarize my session, which are in part seconds. So you have learned the macrophages and neutrophils are specialized the phagocytosis. Macrophage and neutrophils. So these are the very important cells which are available in our body systems, which are specialized. They can phagocytize the antigens or tumor tumor cells are virus infected cells. They get phagocytized with the help of the macrophages. Another cell which are very important, that is known as the dendritic cells. Dendritic cells play the important role in the activation of the T helper cell along with the macrophages. So they will activate through the macrophages, and T helper cell along with the macrophages ultimately they will kill the pathogens. The lymphocyte are differentiated into B cell. I have already discussed it, the lymphocyte which are differentiated into the B cell. So their lineage I have discussed. In this case, the B cells, it will generate into the generate through the hematopoietic stem cell, or ultimately it will convert into the B cells, plasma cells and memory cell. And plasma cells, they are able to generate to the antibodies, and T cells they can recognize through the MSC class first and class seconds. And another is the natural killer cells, and based on through the presence and absence of the specific membrane receptors are there. And B and T lymphocytes are the central cell of the adaptive system. So you can say the your a number of the cells are there. They are available for the innate immunity, and another is the adaptive immunity are there. So T cell, B cell and NK cell which are involved in the adaptive immunity, but other cells are there like macrophages and monocytes which are involved into the adaptive humoral immunity are there. So dear learner, I have discussed the various things related to this topic. You have learned the number of the things like hematopoiesis, how hematopoiesis will take place, and how the cell differentiate into the different type of the cells are there, like myeloid lineage and lymphoid lineage. You have learned it. And another thing you have learned how to different transcription factors, or you can say the growth factors, which are involved in the differentiation of the various lineage, like myeloid lineage and lymphoid lineage are there. In case of lymphoid lineage, it will convert into T cell, B cell, and further it convert into the plasma cell and memory cells are there. So you have learned number of the things related to this topic. So dear learner, you have any problem related to this topic, you can mail me at crm1@ignou.ac.in. If you want to a detailed study, you can go through the IGNOU SLM BZYCT 141. It is the immunological courses. You can go through the unit number 5. You can clarify each and everything. Thank you.
Hello learners, in this program I will discuss about the Rapa-Kivi texture. It is a magmatic texture in which alkali feldspar is rimmed by plagioclase. Remember, alkali feldspar is rimmed by plagioclase. Plagioclase is calcic plagioclase. Example is Rapa-Kivi granite. Rapa-Kivi texture found in granite, therefore we call it as Rapa-Kivi granite. We use Rapa-Kivi texture as a prefix here to define a rock. Rapa-Kivi granite shows feldspars. These are the feldspars, rounded feldspars. These are the rounded feldspar grains without plagioclase rims, and typical orthoclase feldspar with darker plagioclase rims. Some of the orthoclase they have darker plagioclase rims. These are the darker plagioclase rims here. As a result, you get formation of Rapa-Kivi texture. How this Rapa-Kivi texture looks under the microscope? It shows Rapa-Kivi texture. Here Sanidine is a high temperature feldspar, rich alkali feldspar surrounded by polysynthetically twinned. This, see here, these strips of polysynthetically twinned oligoclase. Oligoclase is again a variety of plagioclase. So, these feldspars
Namaskar. I welcome all the students of PG Diploma in Food Safety and Quality Management Program and the MSc in Food Safety and Quality Management Program of 2023 January session to this session on Introduction to PGDFSQM and MSc FSQM. As this is your very first teleconference session for the current batch, so today I'll discuss about the basic features of these two programs, the syllabus of these two programs, and what you are going to learn in theory and practical, and what are the different support activities you will be receiving from IGNOU, and what are the evaluation methodology for these two programs. So let's begin with the MSc program. I am discussing today two programs together. One is PG Diploma, another one is MSc. Why? Because these two are closely related. MSc FSQM program, that is MSc in Food Safety and Quality Management program, is a modular program. Why? Because this is having multiple entry and multiple exit option. So there is one exit option after 1 year of completion, successful completion of the MSc program. That means if anyone who has taken admission in MSc FSQM program, if he or she wants to discontinue the program after completion of first year, the person can exit with a PG diploma. The person will get the PG diploma in Food Safety and Quality Management. So that is the exit option after 1 year. There is one option for lateral entry also to the MSc program. The person who has already completed PG diploma in Food Safety and Quality Management, that person can take admission directly into the second year of the MSc program. So that is the lateral entry option for the MSc FSQM program. The person need not to repeat the first year syllabus. So the first year syllabus of MSc FSQM program is same as the syllabus for the PGD FSQM program. So I'll discuss both this program together today. If you see the objectives, what are the objectives of the PGDFSQM programs, what you are going to learn from this program. So I'll say that the major objective of this PG Diploma program is to develop the professionals who will be able to develop, design, implement and audit the food safety and quality management in the food industry and in any food establishment in the country. So how will that be possible? Through this program, you will learn to understand the issues of food safety and quality management in the food production chain. Food production chain, I mean to say the starting from food production, food processing, food packaging, food retailing till consumption throughout the food chain. So you will be able to understand the issues related with the food safety and quality management. Then this will also help you to build your technical proficiency in undertaking the food safety and quality assurance activities throughout the food chain. This will also help you to undertake the microbiological and chemical analysis of any kind of food product. You will also be able to apply GMP, that is good manufacturing practice, good hygienic practice, good laboratory practice, good transport practice, then HACCP, that is hazard analysis and critical control point. All these good practices you will be able to apply after going through this PG diploma program. You will also be able to effectively plan, conduct, report and audit any food safety and quality management in food processing or food production, food retailing, means food establishment as per the guidelines of the ISO standard, that is 19011. So these are the objectives of the PG diploma program, and definitely these are also the objectives of the master's program. But as you know that master's program, there will be a second year course also. So we learn something more. So what are those courses? Those courses will help you to learn about the food toxicology, the public health, the emerging issues, biotechnology, the research methodology, and definitely you will be doing one research. So the objectives of master's program are to develop the competent human resource in the area of food safety and quality management for different food stakeholders like the food industry, like the food regulators, the research and academic institutes which are dealing with the food, the accreditation and certification bodies, the food testing laboratories, food training centers, and the students will also conduct the research studies in emerging food safety and quality issues. So now we'll see the duration of the program. For PG diploma program, the minimum duration is 1 year, maximum 3 years. That means you can complete your PG diploma program within 1 year of your registration. If by any chance you fails to complete in the first year, then you can complete the program within 3 years of your registration. That is the maximum duration of your registration. Then the second thing is the MSc FSQM program duration, minimum duration is 2 years. You can complete the MSc program in 2 years time. If you cannot do it within 2 years, your registration will be valid till the 4th year of your registration. For the MSc student, my request is to please re-register yourself after the completion of first year for the second year of the MSc. And the medium of instruction is English only. Now I'll show you the program structure of the PG diploma and the MSc program. So as I mentioned that the first year syllabus of the MSc and PG diploma are same. So this is the syllabus for the first year of the MSc and the PG diploma. In first year you will study 36 credits of courses. There are total 8 courses. You can see here that 4 courses are completely theory based. There is one course, second course, that is food microbiology, that is having theory and practical both the component, that is an integrated course. There are 2 completely practical based courses and there is one project work. So I'll discuss about all these courses just later on. Let's see the second year course for the MSc program. And again there will be 36 credits courses in the second year of the MSc. There are 5 theory courses and 1 seminar and one dissertation, and at the last you can see that there is one project work also for those only who have taken admission as a lateral entry to the MSc and who has completed only 32 credits in the PG diploma program. They have to take the 4 credits and the extra project work in the second year of MSc. So now I'll discuss the courses. The first course is about the food fundamentals and chemistry. MBP1 is the course code. Please note the course code. You have to mention it in your exam paper, also in your assignments. You should know the course codes for all of your courses. The first one is MBP1. So you will learn about the fundamentals of the food science and the chemistry. So you can see there are 4 blocks. Means each course will be discussed under few main themes. So those we call as block. So block 1 will help you to know about the different sources of the food, whether it is animal source, plant source, and very basics of the food science. Okay. Then you will learn about the food chemistry. What is the composition of the food? Means carbohydrate, protein, fat, vitamin, minerals, enzymes, whatever present in the food. Then you will learn about the food analysis, that is the theoretical part of the chemical analysis of the food. And fourth block will help you to know about the basics of the food processing, whether it is minimal processing, whether it is primary, secondary, tertiary processing, and definitely the preservation by chemical methods, by physical methods. So you'll learn about all these in the first course. This is a theory course. Second course is the integrated course, having theory and practical both the component. So this is about the food microbiology. So there are 2 blocks only. So you will learn what are the different types of microorganisms present in the food, what are the sources of the microorganisms by which they come into the food, how they can badly affect if we consume the food which is contaminated with the microbes, so the harmful effects of the microbes in the food. And you'll also learn about the beneficial effects of the microorganisms in food industry. For example, for beer production, for bread production, for yogurt, curd production, we need the help of the microbes for the processing of the food or preparation of the food. So those are the beneficial role of the microbial organisms you will also learn in this program. Then you will also learn the theoretical part of the analysis, microbiological analysis of the food here. As this is an integrated course, so you will do the practicals on microbiology also. You will know how to collect the sample for the microbiological analysis, how to culture that for microbiological analysis, what are the different types of media. You will be also preparing media for microbial growth. Then you will also estimate the microbial load for some specific pathogenic organism for like the Salmonella, Staphylococcus and E. coli, etc. in the practical. Now in the next course, that is the third course, is MPP2. This is about the food laws and the standards. So laws and the standards, this course will help you to know about the national and international standards, rules and regulations which are related with the food. In our country, you must have heard about the Food Safety and Standards Act, rules and regulations, and about the authority, that is the food regulator, Food Safety and Standards Authority of India. So you will study all the food regulations, rules and regulations related with food. What are the different regulations any food business operators in our country needs to follow, means which are the mandatory regulations. You will study about those mandatory regulations, mandatory standards. You will also learn about the voluntary standards like the BIS, Bureau of Indian Standards, EGMAC standards, etc. in this course. And definitely you will also learn about the export laws, import laws, and some of the international food standards also in Codex Alimentarius, WTO implications, and different international standard organization standards. Then in the next course, you will learn about the principles of food safety and quality management. Because as a food safety and quality professional, you need to know how to prepare, how to develop, how to implement the food safety and quality management system. So for that, you need to know what are the principles behind that food safety and quality management. So this program, this course will help you to know the basics of food safety and quality management, the concepts of food safety and quality assurance, quality management, total quality management. You will also learn how to do the risk analysis. So what are the different type of food hazards that can be present in food, whether it is microbe, whether it is chemical, whether it is physical agent. So how those risk analysis has to be done, how the risk has to be managed, how the risk has to be communicated to different stakeholders. So you will learn about all this in the theory part of this course, and you will also learn what are the principles of hazard analysis and critical control points, how to develop a HACCP system and how to implement it. And you will also learn about other good practices like good agriculture practices, good animal husbandry practices, good manufacturing practices, good hygienic practices, good laboratory practices. So we will learn about all these good practices in this theory course. The next course is about the food safety and quality management systems. So here you will learn about the accreditation and auditing of any food safety and quality management system. And you will definitely learn about the ISO International Standard Organization, ISO standards. One ISO standard 9000 is for the quality management system. ISO 22000 is for food safety and management system. And you will also learn about the laboratory management system, environmental management systems, and few others retailer standards like IFS, like BSC, etc. Then comes the practical course, MPPL1, and there is another one is MPPL2. So MPPL1 you will be doing some practicals on food safety and quality auditing. So you will be visiting one food industry. You will identify the gaps in the food safety and quality management system for development of HACCP. You will learn how to prepare how to identify critical control points, how to prepare the HACCP system, how to implement it, it's what to do in case of traceability studies, what to do in case of recall. Then you will also do some practicals on the packaging labeling regulations related with food safety and standards regulations. You will compare the packaging of the product with the standard regulation. You will be also doing some practicals on mock audit of any food safety and quality management system. So you will know how to plan for an audit, how to conduct the audit, and how to write the report of the audit. So you'll be doing all these in your practical. Then there is another practical that is on chemical analysis and quality assurance. So there are 28 experiments. So you'll be doing the experiments to know how to estimate the moisture content, protein content, fat content, ash content, mineral content in any food. You will also be doing some practicals to know the quality of the fat, the saponification value, then many other things you will be doing, and you will also be using some of the high-end instruments to know the pesticide residues in a food product, to know the heavy metals residue in a food product, to know the vitamins content in the food product, and you will also be doing some of the sensory quality evaluation of the food. Then there is one project work in the first year of the MSc and definitely in the PG diploma program that is very important. So you have to choose your project topic. So please read your project guidelines which is given in the program guide. The program guide of the PGDFS QM is already uploaded in the IGNOU website. Please visit the website and read it. Probably you have also received the hard copy of the program guide. So read carefully the program guide. You will see that you have to choose a topic. There are a list of suggested topics given in the program guide. So you can choose any other topic also, but it should be related with some activity related to food safety and quality management. So first thing you need to do is to identify your supervisor, project supervisor, who will guide you to do your project. Choose the topic, select your supervisor, write your project proposal, submit it to the regional center. So after the approval from the regional center, you can work on your project, you can complete your project, write your report and submit it to the regional center again. So that will be evaluated by the regional center. And for report submission, there is one deadline. So as you have taken admission in 2023 January session, so last date for your report submission for term end examination December is the 30th November of this year. If by any chance you missed 30th November this year, so you can submit the project report later on also, but if you submit by 31st May next year, then your result of project work will come along with the result of the term end examination, which will take place in the June next year, June 2024. So those are all about the first year courses of the MSc program and the PG Diploma in Food Safety Quality program. As PG Diploma is only of 1 year duration, so for the PG Diploma students, that is the whole syllabus for your PG Diploma program. For Master students, you have to re-register yourself in the second year of Masters, and you will learn the other courses, 5 theory courses in the second year. One is the food toxicology and public health. So you know what are the different types of food toxins that can be present in a food, whether it is from plant source like the mycotoxins, various kinds of poisonous mushrooms are also there, whether those are from the animal source, that can be from shellfish or some other parts. Means the toxins can also come from the processing of the food, packaging of the food. There can be some man-made toxins also, because we use so many pesticides, or we are using so many plastics, so that can also contaminate the food. That can be a toxin in a food. So you'll know about the different sources of toxins, their toxicity in the human body, how they get absorbed in the human body, what are their bad effects in our body. So you will learn about all these in the food toxicology course, and you will also learn definitely about the public health, as it is related with the food consumption. If the food is not safe for human consumption, if the food is contaminated, then it will cause foodborne diseases. So how the foodborne diseases gets transmitted from food to human being, you will learn about that, and how the foodborne diseases affects the human health, you will learn in the public health course. Then there is another course is on food biotechnology. So you'll know what are the basics of biotechnology, how the genetic engineering is utilized for the production processing of the different types of food products, how the biotechnology is applied in case of food fermentation, in case of food production, like for different genetically modified food like different tomato, potatoes, soybean, etc., which are not allowed in our country, but those are allowed in some of the advanced countries. So you will know about the application of biotechnology for the food production. You will also know the advances of food biotechnology. And also the challenges in food biotechnology. As I mentioned, that genetically modified or modified organism or genetically modified plants or foods, what are the challenges in our country for production of genetically modified food. So you learn about all this in the food biotechnology course. Then there is one course on emerging trends in food technology and safety. So here you will learn about the advanced technologies like the membrane processing, like the irradiation, ultraviolet application, ultrasound application, high pressure processing. There are so many novel technologies, emerging technologies which are being used in food industry for production, for preservation, self-life extension, for packaging, etc. for the food. You'll also learn about emerging trends in food microbiology, the technological advances in food safety like the biosensors. Nowadays, biosensors are being used to evaluate the quality of the keeping quality, storage quality of the food. For many other purposes, biosensors are being used in the food industry. You'll also know about the advances in food analysis, how without destroying the food, how can we measure the quality of the food. So that is the non-invasive analysis of the food. So you learn about all these in this emerging trends in food technology and safety course. And definitely there will be also challenges just like the food fraud, food adulteration, etc. Those will also be discussed in this theory course. Then there is one course on research methodology. This course is definitely going to help you to do your research. As a part of your MSc program, you need to complete one dissertation or research course. So for that, you need to know the theory of doing any research. So what are the different scientific approaches you can follow for doing your research? Then what the different research designs which you can adopt in your doing your research? What are the experimental design? What are the tests and measurements you are going to use in your research work? How to write the research report. So you will learn about all this in this theory course. There is another course, this is the theoric course, but this is a MOOC course. MOOC means the massive open online course. That means this is offered in the SWAYAM platform by IIT Kharagpur. So you have to register yourself in this MOOC course. This is offered in January session. So the students of MSc in your second year, please register yourself in this MOOC course and you complete your MOOC course there. You get the marks, get the grade and request the IGNOU for your credit transfer from that to from MOOC course to your MSc program. And in this course, you will learn about the novel technologies like the high pressure processing, the modified atmosphere packaging, active packaging, micro encapsulation, and then supercritical fluid extraction, food extrusion technology, etc. Then there is one seminar component. So this is compulsory for all the students of MSc. In second year, you have to present one seminar and the seminar topic should be related to any of the issues in the food safety and quality management. You have to make an oral presentation in your study center or regional center as you will be directed by the regional center or study center. Then there is a dissertation which is of 16 credits. This is very, very important. So you will have to choose one topic which is related with food safety and emerging issues in food safety and quality management. So you'll be doing your research work either in the study center, in the food processing or testing lab or in a food industry. And you have to prepare your report and submit it to the study center or the regional center. Then for completing your program, for learning all the theory, practical and completing your MSc or PG diploma program, what are the different types of support services the university is going to provide you? These are the first thing is the study material, self-learning study material. I hope all of you have already received the hard copy of the study material. If you have opted for the hard copy, Otherwise, you can access the soft copy of the study material which is available in the EGYANKOSH, EGYANKOSH website. You just go there and search for the PGD FSQM or MSc FSQM program. You will find the soft copy. You can also use the IGNOU e-content app for the soft copy. In the program study center, you will be provided with theory counseling, practical counseling. Theory counseling is, attendance is not compulsory, but for practical, you need to attend at least 75% of the practical, because it is very important to develop your skill. You need to have some hands-on experience, because when you will be joining any food industry or any food testing lab or any other work placements where you need to apply your theory knowledge as well as your practical skills. So please attend all the practical sessions, and the 75% attendance is compulsory. You will be doing your assignments and submitting it to the study center. Then there will be online counseling sessions also, which will help you to learn your subject. And there is one web-enabled academic support, that is, we call it WEAS, WEAS. You can visit the WEAS site, you can register yourself, and you will find the soft copy of the material, soft copy of your program, and there is discussion forum also. You can discuss you can get the clarification of your queries there. You will also find the audio video programs which are available in this program. There will also be IRC, that is interactive radio counseling, and TC, that is teleconferencing, just like today's program. So whenever we plan any IRC or TC, we send you the email. Please attend all the IRCs and TCs, that will help you to learn your subject. And there is definitely be some of the audio video programs which you can use. Those are available in the IGNOU website and also available in IGNOU YouTube channel. Then about the academic counseling, I told you that there will be theory and practical counseling in the study center. In the theory counseling, there will be 2-hour session for theory, and there will be 25 theory counseling in the first year of PG diploma and masters, and the second year also there will be 25 theory counseling. In the first year of your masters or PG diploma program, you will have 50 practical counseling sessions, each of 4-hour sessions. And I already told you that 75% attendance is compulsory. Otherwise, you will not be allowed to appear in the term and examination. Then if you see the evaluation methodology, the theory and practicals are evaluated separately. So theory is having 2 components again. One is continuous assessment, that is your assignment. So assignments for your current session is already uploaded in the website. Please download the assignment for PG diploma program or the MSc program. Write the answers and please read the instructions carefully. So write everything, whatever is mentioned there, and submit it to your study center. Please keep a copy of your assignment along with you. If any case, if your assignment gets misplaced, then you can use it. Then the weightage for the assignment is 30% and the term and examination is 60%. So you have taken admission in January session. First time you will be appearing in the term and examination December 2023. So weightage is 70% for the term and examination. And for practical also, there are 2 components. One is guided practical, another one is unguided practical. So guided means the counselor will help you to do the practical. So that is your regular practical counseling session, that is guided practical. Weightage is 70%. And unguided practicals means that is the practical examination. That means no one will help you to do your practicals. So the weightage for the unguided practical is 30%. And the project work will be of 100 marks. So you need to score at least 40% on individual component and also on aggregate basis. So 40% is the passing mark. Then I'm just sharing with you some of the contact points. If you find any difficulty during the program, while you are doing your MSc or PG diploma, you can, if you have not received your study materials, you can contact the regional center or the material production and distribution division, MPDD-IGNOU. For theory or practical counseling, if you find any problem, so please be in touch with your study center, they will help you. For project work, please be in touch with your project supervisor and the regional center, because you have to submit your project proposal and the report to the regional center. Then any issues related with your examination or results, please contact Student Evaluation Division, IGNOU. For audio video materials, if you want to purchase, please contact EMPC. For any kind of student grievance, you can go to Student Support Services or Regional Center, or you can lodge your complaint in IGRAM also. For any doubt related to your academic content related to a PG diploma or MSc program, if you have any doubts, you can contact the program coordinator of PGDFSQM or MSc-FSQM School of Agriculture IGNOU, and the email is already given there in the program guide in the website, and you can also note it down. It's pgdfsqm at ignou.ac.in, and you can note down the phone number also. So today we discussed about the basic features of these two programs, MSc in Food Safety and Quality Management and PG Diploma in Food Safety and Quality Management program. The syllabus I discussed in details, and the evaluation methodology and the support services you are going to receive from the university. If you still have queries, you can write us email, you can contact us through phone also, but my request is please read the program guide carefully, read the project guide carefully, be in touch with your regional center and study centers, that will help you, and please attend all the counseling sessions, all the theory and practical counseling sessions in the study center. So thank you for listening, thank you for watching the program. Namaskar.
We wear clothes on a daily basis, but have we ever wondered, where do our clothes come from? Who makes our clothing? And who are the people and the process responsible for the whole manufacturing of our clothing? If you want to know more about this, Indira Gandhi National Open University from the School of Continuing Education has launched a program which is Certificate in Apparel Merchandising. There are many benefits after doing this program. You can become a designer in various fields. It can be fashion designer, apparel designer, costume designer, or even a textile designer. One can also work as a fashion consultant. Research and development is another field which one can take up after doing this certificate course. Going into academics is another perk after doing this program. Getting into fashion houses or fashion studios as an intern is also a very good option post doing the certificate in apparel merchandising. The basic eligibility of the program is 10+2. The duration of the program is 6 months to 2 years. The program is also available for both the admission cycles, which is July and January. The exams are conducted twice a year, in June and December. So what are you waiting for? Join this program and get an enriching knowledge in the field of apparel merchandising. And to do so, you can log on to the official website of the university, www.ignou.ac.in.
Welcome viewers. Today we are taking an important topic that is inventory control techniques, which is a part of your course MWR01, that is Introduction to Inventory Planning and Warehousing Systems. The name of the program is Postgraduate Certificate in Inventory Planning and Warehousing Systems for Engineers. When we talk about inventory control techniques, first of all, let us see what we understand by inventory. inventory starts from your house only. You just see your kitchen, you just see your wardrobe where you keep your clothes, that is also an inventory. Now let us come to your kitchen. You will appreciate that you will not go for 250 grams of potato every day, or 250 grams of flour every day, or 10 grams of or 10 milligrams of salt for purchasing. So what do? You keep a stock of it. And if I say in a real word, inventory is a stock of item which is kept in anticipation for future use. So if I say that how we are organizing today's session, we will first give you introduction, then objective, and then finally we will discuss inventory control techniques. Here I just want to tell you that these 7 to 8 inventory control techniques, they are very simple and very useful to you. Whenever you want any project, develop any project in the area of inventory planning and warehousing, you can use one technique or in combination of two techniques. You will find that your project will be completed within no time, and you will have an objective and motivation for doing that particular project. Now, why we are focusing on inventory management or inventory planning and warehousing system? There must be some reason. There are so many examples. I was first thinking to have the name of the company, but instead of giving the name of the company, I have just quoted two things. First thing, you must have heard of many companies who are suffering financial losses, even the loss of brand image, just because of inventory management problem. And Nike, Target, these are the name of the company which stored huge amount of inventory during COVID period. There are two types of company. One company was not able to utilize their inventory, and other was in a stock out condition, and they do not have the plenty of inventory for producing the goods or products. So these both situations are typical and dangerous also, because in both cases you are unable to satisfy the customer demand or customer expectation. So these are the two examples I have given to you. One, where there is a huge inventory, and second, where is a stock out condition. So in both cases, your poor inventory management will affect the performance of the company. So if I say what are the learning objectives for this today's session, you will be able to describe inventory, you will be able to define various objectives of inventory control, and the most important is that you will be able to apply the inventory control techniques. In this session, we will have first discuss what is inventory or what is meant by inventory. As I told you that inventory means a particular items, goods, they may be raw material, they may be work in process, or they may be finished good. Instead of saying these types, you will have another 3 or 4 types of inventory. First is your raw material, then work in process, then finished good, and the fourth one is repair and maintenance, or the maintenance, repair and operations inventory. Because in every organization, whether it is a service sector, whether it is a manufacturing sector, you need an inventory. And for running the production along with the raw material, you need certain tools, certain coolants, certain lubricants for carrying out those production process. Because when the machine fails, what you need? You need a certain equipment, you need certain tools so that you can repair the machine, so that the production process gets started. Another type of inventory is named as a decoupling inventory. Let us take an example of 3 or 4 work center. Suppose A, B, C and D. The item or the component after processing at A, process at B, then after B it goes to C and finally D, and then from D it is in the finished product. Now suppose the machine A is not working. The production at B will be halted or stopped, production at C will be stopped, production at D will be stopped. Suppose we keep an inventory of items produced by work center A, produced by work center B, produced by work center C. So what will happen? If the machine A is stopped, and it will take some time to come out from a breakdown condition, and that time the processing at work center B will not be stopped. Same thing is true with the C also and D also. Now here we have given you a brief introduction of inventory. As I given you one example of your house, similarly there are so many other examples where you can define the inventory. Now the value of materials and goods by organization, there are 3 main points why we need the inventory. We need the inventory to support the production, because raw material is coming, sub-assembly is ready, work in process material is there, so you need to support the production. Similarly, we need the MRO, maintenance, repair and operation inventory, because we want to have the support activities. Then finally we need the finished goods and spare parts or merchandise for the sale and the customer service. Now what are the prime objective of keeping the inventory? First thing, we do not want any disturbance in the production schedule. We want the material should be available at right time, at right quantity and at right place. Second important objective is to keep down the capital investment. Now what is capital investment? Because if you are keeping a huge inventory, that means your capital will be tied up. Other objective is that because we have a semi-completed items, just like work in process inventory, so we should have, as I have given you one example of 4 or 5 work centers where we have decoupling inventory and we don't want disturbance at work center B, C or D. So that is also one of the objective, that there should not be any disruption. So these are the few major objective of the inventory control and management. Now let us come to the main inventory control techniques. I have pointed out and the list is given: ABC analysis, VED analysis, SDE analysis, HML analysis, FSN analysis, SOS analysis, and XYZ analysis. Definitely we are not going to give you the ABC analysis, define analysis with the help of some example. But in today's session, we are only giving you a brief idea about these control techniques, so that whenever in the next session, whenever we take one example, you should recall, or you may recall, what is ABC analysis, what is the VED analysis. So let us first take ABC analysis. If you just see ABC, that means always better control analysis. That is called the ABC analysis. It is a inventory control techniques which is widely used across the industry. Now in ABC analysis, we categorize the inventory into three categories. One is A category, another one is B category, and third one is C category. As the name implies, A category, that means very important, where the value of the inventory, this class of inventory items are very high priced. They are valuable, but they are less in number, but they are very expensive. Now come to the B class or B category. They have a moderate number and moderate value of the items. So that is why they called as a B category. Now come to the third category, that is called as a C category. Under C category, we have a high number or large quantity of the items, but they require lesser investment, or in other words, the value of these items is less. Now let us see this figure. You just see that A class inventory number of items is less, but they consume a huge value of the money. B is a moderate, while C is a large quantity, but the value of the items are less. Now, why we are discussing it? That means the management has to focus, give more importance to A class inventory and less importance to the material which is bulk in quantity as the C. So that is the ABC analysis, and here again I am repeating A class, B class and the C class inventory. Now second inventory control technique is VED analysis. V stand for vital. These are those items without which there will not be any generation, or there will not be any production, or you can say that if this items under this category is not there, the production will be stopped or it will be largely influenced. Now come to the another definition. The three categories are there in which we classify the item. The figure is in front of you, that is again, and this type of classification provides the management a key learning insight. If you just say vital, vital means similarly that the production will get stopped. Under essential, the production will not stopped, but the procurement of such item is very costly. Under desirable item, the production will not stop, but if there is a stock out condition, then the production plan may be influenced or affected, but you are not going to have any danger of the stoppage of the production. So, as a management, you need to classify and what is the use of this VED analysis, because you want to identify that which are the item which are vital in nature, which are the item which are essential category, or which are the item under the desirable. So, essential items, when we talk VED, so essential items, they will reduce the equipment performance, but they are not move towards the inoperative of the system. If there is a non-availability of essential item, there may be a loss or separation of generation work, but it will not lead to the stoppage of the work. Substitution is always there. It can postpone the things, but with the brief repair, we can put the production on the right path. Even the cost brought about this is not very high, while in case of desirable item, definitely it will be very low cost. So, we will see that why it is so useful, because we are categorizing the items into three categories, that is vital, essential and desirable. Now, let us come to the third technique, that is called as a SDE technique. S stand for scarce, D stand for difficult and E stand for easy. Now, where we use this control techniques, we ask the question that whether there is a non-availability of items, how scarce is it, does it have longer lead time? If I again repeat, lead time means the time which is spent at the time of giving order and the receiving of that item as called as a lead time. Fourth question is, where is the geographical location? It is close by or far away, or how reliable are our suppliers? That means if we ask some supplier that we need some item within 2 hours or within 2 days, whether those suppliers are ready or they can be relied that they will give us the item within 2 days or 2 hours. So, now you just see that under SDE how we analyze the situation, that is scarce. That means they are very scarce. It is very difficult. If they are not available, it will take weeks, months or years to get. So, they are called the scarce. For example, if you talk about the power sector, so the flywheels are scarce because of not the, because there are certain government channel through which we need to give order. difficult they are not scarce, but it is very hard to secure or procure them. Third category is easy, where the items are very easy. You recommend and those items will be available. So these are the three categories: S, D, E: scarce, difficult, and easy. Now another control technique is also called as a HML. H means high, M means medium, and L means low. This HML analysis is similar to ABC analysis. The only difference is that instead of users value, we use the price criteria. Now let's see how we do the HML analysis. Please remember, this is only an example. If I say 500 does not means only 500, it depends or it varies company to company. Now suppose a company has items which are above 500, then we say as a high and we categorize under H. If the price of item is between 50 and 500, then we will categorize as a medium and we give a symbol M. The items whose unit price falls below 50, then they should be categorized under low, and the symbol is L. So this is called as a HML analysis. Another control technique is FSN analysis, where F stands for fast moving, S stands for slow moving, and N stands for non moving. Now, which are the items which are fast moving, and which are the items which are slow moving, and which are the items which are not moving? If you just visualize and recall your recent visit to any company, you will find that there are several items which are fast moving. They are issued from the store various times or at more than 3 or 4 times in a day. While slow moving items are those which are issued once in a day. They are issued once in a day. While non-moving items are those which are issued once in a 6 month or once in a year. So this is the beauty, that why we do the FSN analysis, because we want to know that which are the item which are non-moving, which are the item which are slow moving, which are the item which are fast moving, because here we have to give more concentration to the fast moving item. Now another one control technique is SOS analysis. S stand for seasonal and O and S both stand for off seasonal. Now this type of inventory control techniques basically categorize the items into 2 categories. One is seasonal and another one is non-seasonal or off-seasonal. If you just see, you take the example of fruits, then if the season is between May, June and July, they are seasonal items. But if you talk about in the winter, if you are getting mango, it is off-seasonal. So this analysis helps in identifying the items under the category seasonal or off-seasonal. Another control technique is XYZ analysis. Basically, all the control techniques are basically on one fundamental concept, that you want to categorize the items available in the a company into a different categories. And if you just see the idea, that the basic idea is that one is having the high value, one is moderate value, and one is low value. Whether it is any category, you will find that the basic idea is same in all the control techniques. Now just see XYZ item. X item are also called as a runner items. They have a request at least 10 out of 12 months or more. Means minimum 10 times they will be requested for the procurement. Y items at least 4 times, but not more than 9. Z items are those items which gets the request at least 3 times in a month, in a 12 months. Now we will come to the another technique, inventory control technique, that is called as a cross docking. It is a basically different than the inventory control technique which we have discussed. Under this cross docking, we basically, instead of putting the items in the warehouse, we directly supply from a distributor to the retailer. So we are eliminating the need of warehousing, we are eliminating or we are minimizing the cost of warehousing. That is called as a cross docking. Another inventory control technique is first in first out, last in first out. That means old inventory is sold first or used first, and then the fresh inventory will be used later. Second technique is last in first out. That means the inventory which is coming in the last, that will be issued first. Now in this session, what we have studied or what we have discussed: inventory management is an essential part of every business or in every organization, whether it is a manufacturing sector, whether it is a service sector. In order to have the effective inventory management, there are various costs, just like warehousing cost, inventory cost, carrying cost, ordering cost, cost of obsolescence. We need to minimize it, because when we minimize it, we are improving the supply chain efficiency of the business. We need a good forecasting technique, as well as we need a appropriate inventory control techniques, so that we can efficiently manage our inventory. In the next session, we will take example of ABC analysis, so that you can get a good learning insight about the ABC analysis. Thank you.